So, in this series of 3 podcasts with Vet Record, we will be discussing the complex world of the microbiome, the diseases and disorders that can occur when it is disrupted, what the emerging research is saying, and what practitioners can do to re-establish a healthy bacterial balance in their patients. The series is sponsored by Protexin Veterinary in partnership with the British Veterinary Association. In this episode, we will be looking at the microbiome and how it works with the gut and behaviour. We'll discuss how the microbiome can affect cognition and memory and how that declines in ageing animals, particularly in dogs. We'll also look at where neurological conditions come into the picture and whether there are interventions that can help the practitioner tackle this in their patients. So today we're joined by Holger Volk from the University of Veterinary Medicine at Hanover. Holger also was for many years at the RVC. And Sára Sándor from Eötvös Loránd University in Budapest. Holger's clinical interest and research focuses on neurology, particularly epilepsy, and relation to this podcast, the gut-brain barrier, whereas Sára investigates the genetics of cognition and the microbiome in ageing dogs. So welcome to Holger and Sára. Thank you very much for joining us. So first, if I could perhaps ask both of you, perhaps Sára first, perhaps if you just give us an overview of what we're talking about here as far as memory, cognition and neurology, and what population are we considering? I mean, is this just dogs? Is there other things going on in other companion animals?
Well, actually, I'm involved in the field as a co-author of a research paper where we investigated dogs. But more generally speaking, the link between cognition and the gut microbiome has been investigated in other species as well, including model animals like the laboratory rodents. But in our research team, our main focus were on dogs.
Great, thank you. And Holger, from your point of view, from the neurology point of view, what are we looking at here, and what's a good overview of the topic?
Yeah, I mean in general, I was quite flabbergasted to see the interaction between the gut and the brain. Obviously, there's still the brain is still winning; it has a lot more neurons than the gut. But we definitely have seen changes and done some research where we looked at epilepsy, especially, but also in cognition. But there's less known at the moment, and behavior changes we have seen as well. For cats, there is unfortunately not so much data. I think it's an unexplored field. I think we always start in dogs and then we go to cats. Perhaps we should do it the opposite way, but at the moment, that's how it is.
And I mean, Sára, particularly looking at your research, I mean, how does the microbiome make a difference? What kind of things are being affected by it, particularly when it's disrupted?
Actually, if we look outside of the research which I participated in with my research team, there are many papers which investigated its effects on behavior and on different behavior variables, mostly in dogs. Usually, as Holger mentioned, usually dogs are in the focus among the animals living with us. In our research, we were specifically focusing on aging in dogs and the changes in cognitive capacities in the aged dogs.
And what sort of thing were you seeing? What type of results were coming through?
So, first, one of the interesting parts of our research that we could involve family dogs because very often research is done, especially it was done in the beginning of dog microbiome investigations with laboratory dogs, but we included family dogs. And two main types of data were collected. One is the behavior data, together with the other data about the animals like age and the breed. And we also collected the poop samples from the dogs. it was linked with the behavioral data collection. So after the behavior tests, the dogs were taken for a walk. The researchers were going with the owners and the dogs and quickly tried to collect the poop and froze it in the ultra low temperature freezer as fast as possible because time counts. So that was the methodology in short. And this allowed the research team to look for correlations between the behavior variables collected through the behavioral tests and between the microbiome profiles which were analyzed from the poop samples of the dogs. And the two main findings, at least in this population, which unfortunately did not include as many dogs as would be optimal for a large scale study, but it's not so easy to invite so many family dogs for research projects because the owners need the time, etc. But we could still analyze around 30 dogs. So we found a connection between the age of the animals and the abundance of certain bacterial phyla, especially between the abundance of the age, between the age and the abundance of the Fusobacterium phylum. And actually, it could give us a hint on some interesting thoughts about the dog microbiome. But I think we can get back to this a little bit later. And the other main finding was a correlation between the memory performance in the aged dogs, so how well they could perform in a memory test, and between the abundance of the actinobacteria type bacteria. And this could also have some implications in the general notion that dogs can be used as model organisms for human neurological disease and neurological neurobiology, because some studies found similar connections between actinobacteria abundance and Alzheimer's disease. But so far, at least in our hands, in our research team, we could only report these types of correlations and no other correlations with other bacteria, even if several types of bacteria could be detected in these dogs.
Great. Oh, that's really interesting. So obviously some pointers there between cognition and memory and the microbiome. Holger, I mean, I know that your research team have been looking at sort of diet and the effect on epilepsy. How has the microbiome also come into that research there?
Yeah. So when we I guess we have three main areas. We also looked a little bit at cognition. We looked at behavior changes, which were mainly associated with epilepsy and the seizure control itself. And one of the things I think in general one always has to be a little bit careful, because what I feel like we are very similar at the moment, like we used to be with gene research. At the beginning, we found a lot of mutations, but then we looked in the literature and then thought, oh, there might be some associations, and then we never looked at the functional changes as well. And I think that's one of the things. You might see a difference in the, if you think about a forest, and you might see that there are a couple of different trees, and then you find, that does it actually explain that the forest might do something different than the other forests? I mean, so one has to be a little bit cautious. I think we are in the moment in a very explorative phase. We find a lot of changes between species, between certain conditions, between certain species, and so on. But the effect, I think one has to be a little bit careful about. So what we did is we did some medium chain triglyceride diet trials, and we found that dogs receiving those MCTs, and have now done 3 studies, there are responders to the medium chain triglycerides, and there are some dogs which are not responding. And obviously there was not rocket science that we said, okay, let's see, is there any changes in the microbiome? So do we actually, by doing this, instead of just the effect of increasing ketone bodies by giving those medium chain triglycerides, do we actually also change the microbiome? And guess what? We did find some changes, and if you look at our paper, you will see, that make some suggestion how this could interlink. Yeah, so we found some bacteriaceae. I can't say it in English anymore, but we found something which was very close to some changes which has been seen in rodents. Which so that there's a really great study which was once published in Cell, where they transplanted drug response from rodents to each other, and what they found there was that, I mean, that's at least one of the hypotheses, that Akkermansia was involved in that. So they could have a drug-resistant mouse, yeah, with a seizure disorder, and then they could transplant the feces from the ones which were responsive and to the MCT diet, or the ketogenic diet. It was not an MCT diet, it was a ketogenic diet. And then they could transplant the success. So suddenly the mouse, which didn't respond, suddenly responded to it. And they thought it was due to Akkermansia, and they also found changes in neurotransmitters like GABA. We kind of hypothesized that this is the same we have done now multiple other studies where we also found differences. I think where it becomes more interesting, and then that you asked Sára a bit more, is we did a couple of follow-up studies where we looked at short-chain fatty acids concentration, and there we can see actually differences and changes in behaviors, and potentially also in epilepsy control. And we transplanted did an FMT transplant. That's quite interesting as well. But let me, because otherwise I speak for half an hour, and then I'm sure Sára wants to share some more, and then I can give you something about these two tasters later.
Great, thank you.
Yeah, but I can tell you, Susan, my work is my research is definitely quite shit at the moment. So we do a lot of work with feces. So.
So, Sarah, perhaps we could just pick up on that lipid diet, and I understand there's some work that's been done that's looked at how that has affected, also mimicked sort of aging and cognitive decline. I don't know if you could perhaps just chat about that for a little while.
Yes, actually, to be honest, I'm not an actual expert on the effect of diet on microbiome because I'm originally a geneticist. But generally, what I read in the literature, and sometimes I also participate in education, I see that several research teams try to investigate the link between the diet and especially the inclusion of different fatty acids and lipids and the effect. However, in our current research in which we participate, we did not have the opportunity to investigate that. When we analyzed the data, we also checked the diet of the dogs because it was part of the data collection. But at least in our population, we did not find any connection, any correlation between the diet and between the cognitive capacities, so between the cognitive measures of the animals.
Yeah. I mean, it sounds from what both of you are saying, there's a lot to still find out. So obviously an emerging area for the research here. I mean, where do you think's promising as far as sort of next steps for research and, things that might have, good practical application? Perhaps Sarah, if I go to you first and then Holger.
Yes, actually, for the practical application, the research team I am part of is basically originally a behavior focusing research team. So one of the practical applications, which is not directly linked to the microbiome, is the development of these cognitive tests by which the different cognitive capacities of the old dogs can be reliably measured, even if no complicated test laboratories are available. But although this is practical, this is not directly linked to the microbiome, but probably it could be of interest to some who are looking to cognitive tests. For example, the memory test I mentioned was also validated as a test which can be used to compare the memory capacity of dogs under different circumstances. Regarding the microbiome profiles, it is just a hypothesis of ours, but because of the finding of the high abundance of fusobacteria, generally in dogs, And our especially relevant finding that in the aged dogs, so in the dogs who were older, the abundance of Fusobacteria was lower. And
Yeah.
actually, this finding was in contrast with the human findings, because generally in humans, the opposite is seen in elderly people. So they have more Fusobacteria, and also this type of bacteria is generally linked with dysbiosis in humans. And in other animals where the bacteria profiles are known, it was indicated by the literature that the Fusobacteria are mostly prevalent in carnivores. So our hypothesis, actually based on our finding, because this was a highly significant correlation, is that the Fusobacteria level, so this was on phylum level, so these bacteria are probably having a different effect in the dogs than in humans. And this could be an important implication because it is generally noted that dogs can be highly valuable models to investigate the microbiome and its effects on the neural health, because the dog microbiome is very similar to the human. But some bacteria, because the dogs are originally carnivores, biologically
Mm.
must be behaving differently, at least based on these findings. And these can
Yeah.
have implications for probiotic design for dogs and also for research investigating the effect of these bacteria.
Yeah, that's interesting. Thank you. Yes, 'cause I think there is perhaps an assumption that it's going to be fairly similar, isn't it, across the species? So it's really interesting to hear that. Holger, where do you think is going to be, a fertile area for research coming up in your particular area?
Yeah, let me say a couple of things. So one study we did is where we gave dogs, so drug naive patients with epilepsy, we gave them phenobarbital, and which is a standard drug. And what we found was that the dogs responding to phenobarbital had higher butyrate levels than the dogs without. Yeah. So suddenly we found a change in short chain fatty acids, which I think is, I would have never dreamed actually that say they do anything. So the idea was we started with a study where we wanted to see, first of all, how the behavior really gets affected, because we have reported multiple times that behavior is altered in dogs with epilepsy, and it was never clear, is it actually the drug or is it the disease itself? They get a bit more fear and anxiety type of behaviors. And interestingly, when you give phenobarb, actually that domain gets better. So it's probably the disease which is doing, pushing along the behavioral aspect of the disease itself. And but the interesting thing was that we then found, some minor changes in some bacteria, whatever it's for. But we found interesting changes in the short chain fatty acid concentrations. And the other interesting thing, I mean, we didn't find any changes in dysbiosis. So we work very closely with Jan Sokodolski from Texas A&M, right? He's kind of our microbiome guru. And the other interesting thing is we did another study, and you have to hang in there now, where we said, okay, let's go the next step. I think fecal transplants becomes in a couple of years, probably it already is for some standard of care for certain, if they're non-responsive to diets and so on. And so we thought, okay, let's look at the behavior of dogs with epilepsy, because we know that dogs with epilepsy not only get fear and anxiety, they also get ADHD-like clinical signs as well. They have a problem with impulsivity control, with concentration, with their trainability, and so on. So what we did is we were looking for a donor dog which had a very low score on the ADHD-like behaviors. Um, and then the dogs, obviously, which came for the trial, it was just a pilot trial, but we had 10 dogs which were difficult to control. They were at really high scores. And then the cool thing was, so that, if those dogs in the trial were here and the donor dog was here, Then we did the FMT, so we did it rectally. So nowadays we would do it by poop pills, but at that time we did it rectally. And actually the behavior of these dogs went down to the donor dog. So we definitely changed the behavior of these dogs. Epilepsy control is then your next question. When you do these trials, you always find 2-3 dogs which might respond also from the seizure control, but the owners were in general very happy for the behavior, which was our primary outcome measure. And now the interesting thing is we found again changes in butyrate. And then when you look at butyrate as a molecule, and you might know this, they are very have been studied. All these short-chain fatty acids for gastrointestinal health, especially. Resistant starch is one of those classic things in human diet. So if you cook your potato, leave it overnight, and eat it cold the next day, you will have created a lot of resistant starch. It will boost your large intestines to create those short-chain fatty acids. They have shown that they not only locally might improve intestinal inflammation and so on, but also in general improve inflammatory status in the patient. And the interesting thing from our study was that they could, one of their theories was that there might also be another alternative fuel source. But we also found that dogs increased their GABA. So we looked at GABA metabolism, at neurotransmitter, and it was increased. So how this works, I have no idea. But somehow, by giving them poop, we changed it.
Right.
I would have never thought. So first, I never thought that phenobarb would change your guts in that way, that you suddenly, why would that suddenly shift it? And then you have butyrate, right? And then the other thing is, I would have never thought with FMT you could have such an effect. So we are now doing other studies which are, initially we were not allowed to use. We had to get pilot data because of animal rights, to get to power analysis and so on. So now we are in the next stage. But still, you couldn't doubt it that the behavior changed so much massively. So isn't that amazing? So, and we know this from humans. So this is not, it's enough there. We know that people with certain anxieties and depression, you give it to rodents and they show the same behavior. And we know all that stuff, right? But it's still, I think, absolutely fascinating. And then lastly, and then I shut up again. Sorry if I get excited, but we did another study where we put poop from. So we didn't use the solid part, just the fecal supernatant. And that's published now as well, where we use dogs which were controls, drug naive dogs, dogs which responded to phenobarb, and dogs which did not respond to phenobarb. And we looked at the firing rates of myenteric neurons. And the interesting thing is that they changed. So you kind of look at a couple of matrices, and I'm not a myenteric neuron specialist or whatever, but we work with a very good group here in Hanover, which do normally gastrointestinal disease. But it look at the percentages of neurons firing and at their burst frequency, and it looked like that you could replicate that the poop was changing the firing rate. And it actually, one of the ideas afterwards was like, so if the drug resistance fired differently than the drug responders, so could it be that even already in the intestines via the vagal nerve or via cascades like butyrate or whatever, could modulate what happens in your brain? And we know that vagal nerve stimulation is a treatment for epilepsy, and this is just thinking out of the box. I have no proof for that, but that I was really impressed by this data, because I never thought it would actually change anything. Why would supernatant and whatever it is, who knows? We did find potentially some changes again in short chain fatty acids. That's why we went down this track a little bit, but it could also be just a wrong association. But it is interesting. So something is happening. So
Yeah,
it changed
sure.
behavior, probably epilepsy already on the firing level there, and there's now a lot of research going on in actually stimulating not only the vagal nerve but also other parasympathetic nerves, and seeing if they can change inflammation pattern and so on. It's a big business going on at the moment.
Yeah.
So, but some, I mean, yeah, we are very much, I think, at the beginning of the science there.
Yeah, sounds exciting though, and I mean, I think with the behavioural aspect, obviously that's an element that a lot of practitioners are having to deal with at the moment. So, yeah, it'd be interesting to see. I mean,
Yeah.
from the practitioner point of view, you're saying it's all very early days. Do you think there's anything that practitioners can do at this point to maybe help their patients? And what was it? Too early to
Yeah,
say?
I think it's probably too early to say. I think where we do have some more evidence is for if they have inflammatory bowel disease, IBD, and they do FMTs, which is also depending where you are, and you get this, you will also sometimes see a change in their behavior of the animals. But if that is just because we know that if humans will have IBD or some leaky gut syndrome, they can be quite disturbed by that, and that they can definitely have an effect on your mood as well. If there's a secondary or not, I don't know. I think for our part, what Sára and I do, I think we need probably more data to see if there's really an effect. And then the question will be, can you replicate it? I think they have some good data for the gastrointestinal disease that they can replicate results, but I still wonder, how do we normalize, and how do we know which donor dog is the right one? And obviously, in the first study, what we did is we had an epileptic dog which responded very well to phenobarb. How would it be if you actually take a dog without epilepsy, which doesn't have any behavior issues, and then you would give this as a donor? To get a donor dog, and I mean, there's a lot of specialists in the UK one can ask, but in general, it's not as simple as it might sound. You have to do, check that they don't have any dysbiosis, obviously, that they don't have any parasites and so on, and make sure. And I mean, Jan did check everything for us to make sure we're okay. Saying this, the other thing is what you have to consider as well, and that was happened to ours. We had the donor dog, and then at one stage the lady got a baby, and so she was not as was a bit reluctant to continue getting poop for
Collecting the
us, right?
samples, shall we say?
So we also need to think about the donor's life as well. So yeah, treat them very specially. And somehow the other question we don't know is, some dogs, for gastrointestinal, they have to go there every month. We found in our dogs, interestingly, that actually the main effect was even seen after 6 months and longer. So we didn't have to replicate it. So again, look, as always, when you find something, there's more questions than answers.
Yeah, great. Well, I think that's so the we always have to think of the client, don't we, Holger? So you brought that in there as well. Sára, was there anything you would recommend to practitioners at this point? I mean, I know you talked about, maybe looking at the dog's microbiome, and if people are developing products and things like that, that they're going to be different for people. Anything there that you'd like to expand on?
But actually, regarding the possible recommendations, I think the main message from our study is the indication for the necessity about for the further research on the fusobacteria in dogs, because as Holger mentioned, we always get more questions than answers, and this is especially true for the link between those billions of bacteria and health and behavior. Just an interesting thing, however, it was not published in the final study because this was just one case among the dogs we investigated. But it could be, it was an interesting case because one of the dogs was injured before the second sample collection, because we compared the bacterial composition of the dogs at 2 time points. There was 3 months difference. So we collected poop samples at 1 time point and 3 months later, because we also wanted to get a glimpse into the normal changes in this population of dogs. And one of the dogs accidentally had an accident before the second collection and received antibiotics. And it was actually very eye-opening to see that the sample which was taken soon after the weeks when the dog received antibiotics contained much less bacteria. So the abundance of some type of bacteria was much higher, while some others practically disappeared from the sample. At least they were not detectable by the method used. This was 16 sRNA sequencing. So it was really eye-opening to see that this actually happens when the animals receive some kind of antimicrobial drugs, so antibiotics, that it changes the microbiome fundamentally.
Yeah, that's a good another good reason to be cautious with those antibiotic prescriptions, isn't it?
No, and it goes on for ages, you know. I mean, we all learn to give metronidazole, you know, for certain cases with gastrointestinal disease. You know, we were giving it, I think, like, I mean, it sounds like a bit too much, but like sugar pills, right? We didn't think so much about it, but now suddenly we think like completely different because it ruins your microbiome for a long, long time. You know, if you look at the data, it can 7 to 12 months of not getting back to normal.
Gosh, really? Yeah, so pretty much a year. Wow, that's interesting, isn't it? Yeah. Great. Well, thank you guys. We're sort of coming up to the end of our time, but it's been really interesting. I mean, I think what I would sort of sum up from this is it's a great area of interest to keep looking at, and sounds like it's got huge potential. We just need to know a bit more. I mean, the behaviour element, um, aging and cognition is obviously always going to be an area that people will want to sort of improve for people. And I mean, the things that you're putting out there, Holger, about the 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 change in behaviour being so significant, I think is probably going to be a really exciting area. And obviously, Sarah, your fusobacteria looks like, let's watch this space, see what we get. And I suppose for the practitioners, it sounds also like, just as we're putting out at the end, just think perhaps before using those antibiotics because of the difference it can make, and anything they can do to help, re-establish an animal's microbiome sounds like it's only going to be for the better. So that leaves me just to thank Holger and Sára for the discussion, and so I, for one, will certainly be thinking differently about the gut, cognition and behaviour, and how that microbiome can make the difference. Thank you.
Yeah, you are what you eat, right?
Yes.